Thank you, Dr. Meekins, and thank you to everybody that's here tonight. Fortunately, I'm on Central Time, so it's only about 9:30, 10 o'clock for me. But for those of you that are on Eastern Standard Time or even overseas, I know it's much later or earlier in the morning. So thank you all for being here. Like Dr. Meekins said, my project is titled Renal Mitochondrial Gene Expression is Dependent on Diet in Obese Mice. And I'm in Dr. David Pollock's lab at the University of Alabama at Birmingham. And our lab is really interested in really hypertension, but everyone's looking at various aspects of hypertension. So I've become really interested in the interplay between diet or feeding behaviors and how that can control blood pressure mechanisms or blood pressure rhythms throughout the day. And so this project really is kind of one that I became interested in just off of a whim, reading some literature, but it's really turned out to have some really interesting preliminary data that I'll show you today. And I'm definitely looking forward to kind of fleshing this project out a little bit more and possibly getting a publication out of it. So as an outline, I'll go into a little bit about background, my methods, some of the results that I have, the conclusions we're able to draw from those results. And then towards the end of my talk, I'll talk a little bit more about some of the extracurricular things that I've been doing, or rather, quote unquote, alternative things that I've been doing outside of research. And then, of course, I will take as many questions as you all drop in the chat box. To start, I have nothing to disclose. So by the year 2030, over 50% of US adults will be obese. And we're located obviously in Alabama. And so as you can see here, by 2030, the Southeast will really get hit the hardest by this. And so severe obesity is likely to become the most common BMI category. And this is particularly in women and non-Hispanic Black adults and low-income adults. And so as we also know, obesity is a total body disorder with a number of comorbidities. So a lot of the ones that we generally are interested in our lab are sleep problems, things dealing with circadian rhythm, high blood pressure, cancer, diabetes. But you can see that obesity really lends to a whole list of comorbidities that affect the entire body. So looking at kidney disease, because obesity does cause high blood pressure, which is related to the kidney, we know that obesity is a potent risk factor for chronic kidney disease, and that your intake, the things that you eat, affect the onset and progression of CKD. And so not only does diabetes serve as a potent risk factor for CKD, but a lot of the comorbidities of diabetes lend to CKD as well. And so in particular, as you can see in this picture here, it's kind of is a figure that really illustrates what I've just said. Things that happen that are lifestyle choices, such as diet, physical activity, so on and so forth, can really kick off this feed-forward mechanism that leads to obesity, which is interchangeable with diabetes, hypertension, and that can feed forward into other risk factors for CKD progression, such as proteinuria or sleep apnea or cardiovascular disease. And ultimately, unchecked CKD can lead to end-stage renal disease, where someone would need a kidney transplant. And so we also know is that in the obese state, so here on the right, there's this compensatory hyperfiltration that occurs to meet the increased metabolic demand of having increased body weight. So there's essentially this increase in intraglomerular pressure that occurs as a result of obesity. And so as you can see in this picture on the left at the bottom, we have a capillary leading into the glomerulus that has these podocytes that kind of line it and kind of help to keep larger molecules such as albumin or adipose out of the general circulation. But as you can see in the obese state, the arterial and the capillary start to dilate, which increases capillary diameter, but the podocytes don't increase in size. So you end up with decreased podocyte density because of the increased surface area, and in that way, proteins are able to get out into the urine, which is where you'll have proteinuria, and it also increases capillary wall tension, leading to things like high blood pressure. So we know that obesity, and especially morbid obesity, can induce afferent vasodilation, which augments glomerular filtration rate. So glomerular autoregulation might be impaired, and elevated systemic blood pressures can then be translated to glomerular capillaries, as you can see here on the left or on the right, and this can lead to barotrauma. So this dilation of glomerular capillaries can heighten susceptibility to barotrauma, and then you'll need, of course, more podocytes to cover a larger surface area. So the prevalence of obesity and obesity-associated morbid conditions such as type 2 diabetes, hypertension, dyslipidemia, gout, and hyperuricemia are increasing in both developed and developing countries. And so type 2 diabetes in particular is a leading cause of end-stage renal disease, and it contributes to a heavy socioeconomic burden in developed countries. So to switch gears a little bit, as I mentioned at the beginning of my talk, our lab is also very interested in circadian rhythm. And so when we think of circadian rhythm, generally you'll be told that there are two aspects. One is the central clock, which is housed in the suprachiasmatic nucleus of the hypothalamus of the brain. And this central clock is really just sensitive to light. And you may hear some people refer to it as a Zeitgeber or time giver. This is essentially just the external cue that is able to kind of dictate the expression of genes in the suprachiasmatic nucleus. And so outside of that, and I'll pull this figure up. So here we have the suprachiasmatic nucleus that's receiving light or lack of light through photoreceptors in your eye, and that's feeding forward into these peripheral clocks. And so peripheral clocks exist in essentially every other part of the body. For us, of course, we'll be very interested in the kidney during this talk. What I want you to notice is that peripheral clocks are receiving feed-forward information from the suprachiasmatic nucleus, but they also receive a number of other Zeitgebers, such as food intake or activity, or even some endogenous genetic factors can also affect the peripheral clocks as well. And so it's very important to understand the effect of circadian rhythm as it pertains to the kidney, as it's often shown that those who have irregular sleep patterns or circadian misalignments can also end up having things like hypertension or type 2 diabetes. So to kind of link everything that I've talked about in the past couple slides together, I want to talk about this interplay between circadian rhythm and diet and kidney function. And so what you can see here is that here on the right of the slide, what you can see is that kidney function really follows this circadian pattern. And so in these figures, what we're seeing is that on the left of the figure, it's a normal light-dark cycle where animals are on a normal diet. And just simply inverting the light-dark cycle, so essentially mice are on a 12 hours on, 12 hours off light-dark cycle where from 7 p.m. to 7 a.m. the lights are off, and that's active for mice. They're nocturnal animals. And from 7 a.m. to 7 p.m., the lights are on, which is an inactive period for mice. And so putting them on a normal light-dark cycle, you see that there are these rhythms that are established. There are these peaks and troughs of expression of renin, aldosterone, and corticosterone, which are all kidney-associated genes that can play into the regulation of blood pressure control. What you can see is that if you invert just the light-dark cycle, so kind of just changing their circadian pattern, you really start to see that where there used to be peaks, there are now troughs and vice versa. And so at least for renin, aldosterone, and corticosterone, which are three very important genes, we can see that they display this diurnal rhythm that can be inverted by reversing the light-dark cycle. And so to follow up on that, Dingwo Zhang, who is a recent graduate from our lab, also did some similar work where he saw that just simply moving, not changing the light cycle, but simply changing the time of day that mice are allowed food is enough to completely invert their diurnal rhythms of their blood pressure as well. So again, this speaks to how the timing of food intake can drive the circadian rhythm of your blood pressure. And this is really important because in those who have acute episodes of circadian misalignment, such as those who experience jet lag, or those who have chronic episodes, such as those who do shift work, this really significantly increases your chronic kidney disease risk, as well as a lot of the comorbidities that lead to chronic kidney disease, such as, of course, obesity, hypertension. And so to get really into my work, I became really interested in renal mitochondrial dysfunction in obesity, because looking through the literature, I saw this kind of pattern that high-fat diet in mice is enough to dampen diurnal rhythms and whole body metabolism. And that's some previous work that we've seen in our own hands in my lab. What I also saw is that a lot of CKD models have mitochondrial dysfunction. And so the Asher lab in Israel, for example, has shown that oxygen and carbon dioxide rhythms are controlled by the circadian clock. And in our own lab, we've also seen, to go back to the first point, that putting mice on a high-fat diet for 16 to 20 weeks is enough to induce obesity spontaneously, which coincides with the dampening in diurnal rhythms in their whole body energy metabolism. And so what I also know is that the kidney requires these massive amounts of energy to accomplish reabsorption. So although the kidney is less than 1% of the body's mass, it consumes about 10% of the oxygen that's used in cellular respiration. And so mitochondrial damage and mitochondrial dysfunction are recognized as a major pathogenic effect in chronic kidney disease and acute kidney injury. And so mitochondrial dysfunction, as you can see here, is really an interplay between a number of processes in the mitochondria, any of which can go wrong for a variety of reasons, but can all feed forward to mitochondrial dysfunction and ultimately chronic kidney disease. And so in a lot of AKI or CKD models, mitochondrial dynamics are totally disrupted, which leads to mitochondria being fragmented, having permeable membranes, and also having these increases in reactive oxygen species production. So in obese individuals where they have this compensatory hyperfiltration mechanism to meet the increased metabolic demand of having an increased body weight, you really run more of a risk of having these aberrations in mitochondrial function due to how much more they're having to work. And so looking through the literature again, in some of the previous slides I've shown you, we've kind of established this relationship between mitochondria and feeding or overall metabolism and feeding, between diet and circadian rhythm, but there's not a lot known about really bridging this gap. So not much is known about the relationship between feeding behaviors and renal mitochondrial gene expression. And so that really brings me to my ultimate question for these experiments, which is, are feeding behaviors such as dietary fat content and timing of food intake sufficient to alter renal mitochondrial gene expression? And so to get into my methods here, I had two groups of 8-week-old male C57 black 6 mice. One group of them is on a normal fat diet, which is only 10% dietary fat content. The other is on a high fat diet, which is meant to reflect more of a westernized diet, and it's 45% dietary fat content. So for our high fat diet guys, we put them on their diet for 18 weeks, after which they were all obese. And from there, we were able to split them further off into two more groups that were either fed ad libitum, meaning food's available 24 hours a day. They can come and go to their food bank as they please. And the other half of them were put onto a restricted feeding protocol where food was available rather than 24 hours a day. It's available only during the inactive period of their day. And so we put them on these respective feeding protocols for 2 weeks. And after that, sacrificed these mice, collected their entire kidney and split it up into the 3 functional units of the kidney, the inner medulla, outer medulla, and cortex. And so from there, for these experiments, the data I'll show you is using the outer medullary tissue. And I use this because the proximal tubule and the medullary thick ascending limb of Henle have this really high metabolic rate, which is related to this high mitochondrial content that's needed to sustain the sufficient ATP production to support the levels of electrolyte transport activity in this particular section of the nephron. So I really just use the outer medulla because I know that this is a place where there's going to be a lot of mitochondria, so I figured we would see the biggest effect here. So from there, I was able to homogenize this tissue and create a cDNA library and load them into this really cool tool called a mitochondrial microarray. And so how this works is essentially we ordered actually I'll go into that on the next slide. So to really make it simple, I loaded the cDNA into a ready-made plate, popped that into our PCR machine, and it spits out some numbers that I'm able to plug into a software that comes with the microarray and generate the figures that I'll show you on the next couple of slides. So to get a little bit more into the microarrays, I know that this is generally something that people are really interested in, and I'm pretty excited about it as well. This is a tool that allows us to, using one 96-well PCR plate, look at 84 different mitochondria-associated genes, and the plate also has the appropriate controls loaded into it as well. So it has the primers and everything that you need. All you have to do is add cDNA to CyberGreen Master Mix, and then pipette that into every single one of the wells. So it is a little costly because each plate equals one sample. So you're able to just add your cDNA and CyberGreen into the plate, run it in your RT-PCR instrument, and then, of course, do your data analysis where you can create volcano plots or scatter plots, whichever is more applicable for you. What I really love about this array is that it really is the closest thing to what I would call kind of medium throughput that you can get at a lab bench. So just doing these 84 genes and their controls gives you enough information to look at over 170 mitochondrial associated pathways. And these range from transport, protein targeting, mitochondrial fission and fusion, localization, apoptosis, really spans the gamut. And so it's a really effective tool for kind of looking at a number of genes and then from there being able to kind of pick out what you're interested in. And so to orient you to the next couple of slides I'll show you, which have my data on them, I'll be using this scatter plot to demonstrate my data. And so essentially what you'll look at is on the x-axis is one group, one gene, or one group of mice that have been fed a particular way. And you're looking at the gene expression of one group relative to the other, essentially. And so looking at here the effect of the diet, regardless of time, what you can see, actually we're looking at time regardless of diet, sorry. What you can see here is that the black dots, each dot represents a single gene. The black dots are genes that have been unchanged in terms of relative expression between the two groups. Blue dots mean that they're upregulated and red dots mean that they've been downregulated. And so if you're looking at the normal fat diet at the ZT5 and ZT17 time points, so this would be falling squarely in their active and inactive time periods, respectively. What you can see is that only one gene is upregulated and there are no genes that are downregulated. So if you're looking at the high fat diet at the same time points, where here you have their active period on the bottom and inactive on the y-axis, what you can see is that there are 18 upregulated genes and only one downregulated gene, which really suggests that there's no real effect of time of day on mitochondrial function due to diet-induced obesity. So when we look at the normal fat diet versus the high fat diet, taking time out of the equation, we can see is that there are 23 genes that are upregulated and 39 genes that are downregulated. But when you look at the same thing, looking at the normal fat versus high fat diet during their inactive period, there are 43 upregulated genes and 20 downregulated genes, which suggests that there's kind of more of this differential effect of obesity rather than time of day. And so looking at that data, we became very interested, but we also had some time-restricted feeding mice. So all of these mice were kind of fed ad libitum. They were just on a high fat or a normal fat diet. So we then wanted to throw in our time-restricted feeding mice and just kind of take a look at how that may change the mitochondrial gene expression. And so to remind you again, these are high-fat, time-restricted feeding animals where they're on a high-fat diet. It's 45% dietary fat content, and they're only allowed to eat during their active period. So this would be that 7 p.m. to 7 a.m. And so what you can see here is that when the high-fat diet is restricted solely to the active period, there are 27 genes total that are differentially expressed between their active and inactive periods, which indicates this improvement in circadian expression as compared to the high-fat diet alone or any particular one time point. And so looking at this, we became really interested in this pattern. And so the microarray, as I said, gives you 84 mitochondrial associated genes. And I kind of looked through the data and picked out some of the genes that fell in gene families. So the plate has, a lot of different genes, but it has a couple of very specific mitochondrial associated families. So I picked some of the genes from those families and then kind of tied them back into the function of the kidney and looked a little bit more into those and validated it with qPCR, which is my next few slides. So the first protein family that I decided to look at was the mitochondrial uncoupling protein family, or the UCP genes. And so we know that these genes are responsible for reducing mitochondrial membrane potential, especially in the context of oxidative phosphorylation. So mitochondrial electron transport, for those of you that are very sciencey, you'll remember that it establishes this electrochemical gradient that's used by the F1, F0 ATP synthase to create ATP. However, not all of that potential energy is coupled to energy production. Some of it is lost from protein leak or electron leak. And this process can be induced by uncoupling proteins. So these proteins really function to allow proteins that leak into the inner mitochondrial membrane space to escape into the outer mitochondrial membrane space, which reduces the mitochondrial membrane potential and prevents the accumulation of reactive oxygen species, which can kind of feed forward into a lot of different dysfunctional diseases in the body. And so what we don't know about these isoforms is really the function of each of them. There's some general ideas about how they may function in terms of non-shivering thermogenesis or other, metabolism-related pathways, but there's really nothing known about the particular functions of each individual isoform and whether they're functionally redundant. What we do know is that UCP2 has been linked to hypertension, hyperglycemia, high salt-induced endothelial dysfunction, and it also improves hypertensive target organ damage when it's overexpressed. So it seems like UCP2, if we were to pick one of these three UCP family genes, UCP2 would be the one that's kind of most relevant for us. But because I had all three on the mitochondrial array, I wanted to look at all three of them. And so looking at some qPCR data, what we see is this really interesting pattern where in the normal fat, high fat, or high fat with time-restricted feeding, actually the normal fat or high fat only groups, what you can see is that there's this minimal change in gene expression. However, in the high fat diet fed animals that have had their feeding restricted to the active period, there's this marked increase in expression of all of these genes, which suggests that there's this improved ability of the mitochondria possibly to reduce reactive oxygen species production and manage the increased metabolic demand of the high fat diet and resulting obesity simply by shifting the diet to the active period only. So that was a really interesting pattern, and I wanted to see if it persisted in other gene families. And so another family of genes that I looked at is the SLC25A family of mitochondrial solute carriers. So it's a solute transporter family. It has 53 members, so it's a fairly large family, but it's further divided into subfamilies. And these provide crucial transport steps across the inner mitochondrial membrane. So they provide transport stems for things like amino acids, carboxylic acids, fatty acids, nucleotides, you name it. And the subfamilies are really named based on the solute that they're able to transport. And so SLC25A14 is really associated with ATP transport. SLC25A19 is associated with thiamine pyrophosphate, which catalyzes a number of really critical biochemical reactions. And then SLC25A37 is associated with midopharin 1, which of course is related to iron uptake or heme biosynthesis. And so these are three of the number of SLC25A family genes that were on the microarray. And these were the three that I found the most interesting. So looking at this qPCR data, we really kind of see this same pattern that we saw in the UCP family of genes, which is that in the normal fat or the high fat diet alone, you have this kind of negligible increase in, or this negligible change in gene expression. However, in the high fat time restricted feeding diet animals, you have this massive fold change increase in gene expression for these three genes of this family. And so it was really interesting to see, again, this suggested improved ability of the mitochondria to really deal with some of that metabolic distress that may be happening in the context of a high-fat diet-fed obese mouse. So based on that, we're able to make just a couple of general conclusions. One is that there is no real effect of time of day on renal mitochondrial gene expression, but that time-restricted feeding to the active period may be able to improve the ability of the mitochondria to mitigate some of that increased metabolic demand that can come with increased body weight. And so future directions, obviously I've been presenting this data at quite a few conferences, so I've gotten a whole lot more suggestions than just qPCR and histology, but I definitely want to look at some more of the genes on the microarray to see if this pattern that we've seen in our qPCR data really recapitulates in a variety of genes. But I also want to look at mitochondrial gene expression in the inner membrane, inner medulla, and the cortex of the kidney as well as in extrarenal tissues, so tissues that aren't the kidney, to see if this is a whole body effect or if this is kidney specific. And so that concludes kind of my research associated part of my talk. And science is, of course, a team sport. And so I'm really thankful to have had this group of really talented scientists to do work alongside, especially during the pandemic. So Dr. David Pollock is my PI. And I also have to give special thank you to Dinglo Zhang and Jackson Colson, who's not pictured, who both helped out a lot with the animal work and the tissue collection. And so from there, I'll switch gears to what I was always told was an alternative career, in quotation marks, and that's science policy. So I am deeply, deeply interested in science policy. It's really my career path of choice after I graduate. And so I always, whenever given the chance, like to talk about the utility of science policy as a career choice for those of you who may not feel like research is really what stokes your fire. And so one question I always get asked is, yeah, you want to do science policy, but what can you do with a science policy career? And the answer is literally anything you want. You can work in communication as a journalist or as a lobbyist. You can work in academia and education. You can do advocacy, of course. You can run for office if you want. You can do diplomacy work and work internationally. You can work for the government or for non-governmental organizations. So there's really no cap on what you can do in terms of science policy. It's just very important that you have the knowledge of science to really kind of back up your the knowledge of policy that you'll gain along the way. And so after getting the question of, what are you going to do with this? The next question is generally, what is science policy? And so I generally, to make it very simple, frame it as science writing plus communication plus advocacy. And so it's really working alongside lawmakers to influence policies with a scientific lens. So some of you may have, heard in the past about people advocating for the NIH to receive more funding for research in general or for research in a particular niche of science. And that really is science policy. It's us going to these lawmakers that really often don't have any scientific knowledge and using the expertise that we have, having been in the science field, and lending that to those who haven't been to influence these policies that really will ultimately come back and affect us as scientists. And so this is a picture comic that I've really always liked, is that people always want, no toxins, they want action now, they want a safer world, they want better stuff and things. And for a lot of us scientists, we're like, wait, we want all of that, but we also want it to be science-based decision making. We understand that a lot of it is emotion, we want to save the earth, but we also need to make sure that we're doing it in a scientifically backed way that we know will make sense and will work. And so in the next couple of slides, I'll talk about some of the work that I've gotten into that's related to science policy. And again, this has been just as a graduate student. And so if you're a professor or if you're a faculty member, there's obviously a whole lot more that you can do. So I really kind of got to really kind of flex my science policy muscle during the pandemic. I started out working with the Federation of American Scientists on their COVID-19 chatbot, which was kind of a very early on method of getting people correct information. So I'm sure we all remember at the beginning of COVID-19 when, people were saying that you don't need to wear a mask and, you need to use a hand sanitizer that's 100% ethanol and all of that. And so we were getting a lot of questions such as, can my dog get it? Can my kids get it? Things like that. And there was also a lot of research coming out. And people, of course, were not reading these scientific jargon heavy papers. And so as researchers, there was a massive team of us that were clinicians, grad students, everything in between. And we really took these people's questions, went and did the research, answered the questions, and they were put into this chatbot so that if you were to ask a question like, what % alcohol does my hand sanitizer need? Someone had gone and done the research to show that, 65% plus ethanol content is enough to kill most of the germs on your hands. And so it's just a way of kind of bridging that gap between the public and the scientists who have that knowledge of the jargon, to really be able to answer questions in a fact-based, confident manner. And so that was a really fun project to be a part of. And I think that the New Jersey governor actually ended up using it and implementing it into their state website as well. So it turned out to have a really much bigger impact than we thought it would. I also published an op-ed on biomedical research publishing in the era of COVID-19. Again, a lot of publications were coming out. And so, of course, this argument about open access publishing and how peer review and all of that should go also came up. And so I was able to publish an op-ed about that process and how I could visualize it changing after COVID-19 or even during. And a lot of that stuff kind of persists to this day. People are still having that argument. So it was really fun to be able to put that op-ed together and get it published. Now, pre-pandemic, obviously, so no one has a mask on here, I was able to go to Capitol Hill through the American Society for Biochemistry and Molecular Biology. It was an all expenses paid trip to Washington, D.C., and we spent about 3 days together. First day, we really kind of had this very kind of quick fire training on how to talk to a politician about our research. So someone who probably has no idea about, anything more than basic science, how to distill our PhD level research into something that they can understand. And then we were able to actually go to Capitol Hill and meet with politicians. So this is Rick Allen, who actually is a representative for where my parents live now and where I grew up. And we were able to speak to these people in their offices about increasing NIH funding and NSF funding and really share our own research experiences with them. And it was really well received across the board. So that was another opportunity that I had that was really great. And I'm super excited for the opportunity to go back to DC once things are safe and relatively back to normal. Now, another thing that I had the opportunity to do and do along with another grad student in my program, Adrian Samani, is start our own science policy group. And so the Science Policy and Advocacy Initiative started up in 2019, 2020. And it was really just Adrian and I had this really broad interest in science policy. And we noticed that there were a lot of students who were interested, but they didn't know how to get started. And so we created the Science Policy and Advocacy Initiative in order to host these events where students could talk to people in science policy, where students could kind of come and chat with other students that are interested in policy and come up with action plans for speaking to lawmakers or writing policy memos together. And so this picture is from one of the events that we hosted where everyone wrote postcards to their respective lawmakers, and we actually sent them out. And a lot of the students got responses, obviously the very generic, letter from whomever's office. But it was a really interesting way for people to kind of reflect on the policies that are happening in their local areas or back at home, wherever they're from, and to kind of ask their lawmakers for action on those. So really kind of, again, bridging that gap between the public and scientists and the lawmakers as well. Another thing that I have to plug is the Scholar Strategy Network. I am one of the graduate fellows for the Alabama chapter, and this is a national federated network of researchers in a variety of disciplines. So Pete Jones is one of our leaders, and he is in public policy. Bridget King is in democracy and governance, and then Peggy Biga, who's actually working in DC virtually right now, she is actually in science and technology. And so it's really this repository of researchers who are essentially putting themselves out there, putting their know-how, their expertise out for politicians, lawmakers, for journalists, for really anyone who needs, an expert's opinion to come and ask them questions. And so I think that a lot of states have a chapter. So if you're interested, definitely do go to SSN or Scholar Strategy Network and see if your state has a chapter. And that's a really great way to make yourself useful to, again, lawmakers or journalists or whomever else. And so in terms of getting started, I like to split this up into two things. For anyone, first thing is to talk about science. Even if you have a very basic knowledge of science, if you read a news story that's interesting, talk to someone about it. If you read a news article and there's a paper that they reference, see if you can take a look at it and see if any of it makes sense to you. I would also say that you need to figure out your policy passion. So policy isn't everything. So for me, I'm very interested in healthcare and education, but there are a number of things that scientists can influence policy in. And so it's really important that you figure out really what your passion is policy-wise. Also talk to your lawmakers, schedule visits. If you feel like flying to DC and going to Capitol Hill and knocking on people's doors, schedule a visit or write a letter. You can tag them on social media. There's a variety of ways to talk to your lawmakers, but definitely do that so that your voice is heard. And you'd be surprised at how much more attentive they are when you tell them, I'm a voting constituent for you. And if you want to be reelected, if you want me to tell people to reelect you, you really should listen to what I have to say. Also take part in local groups. I know that in Birmingham, we have a number of policy related groups that are both associated with the university and just associated with the city, but there are always groups that you can take part in. It could be a Google search, it can be asking around, but find those groups. And if you can't physically take part, donate money to them or make yourself useful otherwise. For academics, first thing is always join professional societies. If you are doing anything above graduate level research and beyond, you should definitely be a part of some professional societies. And a lot of them have a policy arm, and they're very happy to pull you in on whatever it is that they're doing. Another thing is to possibly found a policy group. For Adrian and I, that was really a lot easier than we expected. The hardest thing was always finding funding, but you'd be surprised at how many people will throw money at you because you live in the Southeast, particularly in Alabama, and you want people to be more aware of policies that are likely going to affect the people that live there. So found a policy group. And if anyone's interested in this, I'm very happy to offer help. Find a policy mentor. This can be someone at your university who is in a policy related department, public policy or anything like that. Or it can be someone who works in a politician's office, a staffer or a district chair, something like that. Find someone who has policy know-how if you don't, and really kind of link up with them so that you can almost work as a tag team. You'd be surprised at how much you can get done that way. Speak with your university's government relations office. They generally have a number of things going on, and they're generally very open to kind of sharing their experiences with you and letting you know where you can be of assistance. And the last thing, of course, is to read and communicate. There's policy literature the same way that there is research-related literature. And you also, of course, have to communicate what you know. Don't be afraid of using social media. There's a lot that can be done with Twitter or Instagram or even TikTok nowadays. So don't be afraid to communicate what you know and what you're hearing about at any given time. So that concludes my talk. I have to thank my PI, of course. He allows me to do my research and kind of feed my passion at the same time. Pete Jones and Peggy Biga are some of our leaders at Alabama Scholar Strategy Network. Dr. Ben Korb and Andre Porter were really instrumental in getting me to DC through the American Society for Biochemistry and Molecular Biology. AAAS, really great repository for anything science policy related. They have a massive policy arm. And then Adrian and the Union of Concerned Scientists for helping me to found the Science Policy and Advocacy Initiative, and then also offering funding and support. And the National Science Policy Network, the Science Policy and Advocacy Initiative belongs to the National Science Policy Network, so we're a part of their federated network as well. That concludes my talk. There are a number of ways that you can get in touch with me, and I am happy to take any questions.
All right, fantastic. That was a great talk, both on the research side and on the policy side. That's very cool. Please feel free to get questions in as we're going. You're not limited on asking the questions, so feel free to get them as we go. We'll just go ahead and start working through the questions and kind of go from there. So the first one here is, do you know what the podocyte differentiation path is from stem cells?
That's a very good question. I don't know. My research has never been strictly related to podocytes, so I've never looked at their differentiation pathway. But that's incredibly interesting. That would be an interesting thing to look at in terms of why they're not able to kind of multiply and cover that increased capillary diameter that happens in the context of obesity. So the answer is no, but that is definitely something that would be interesting to look into.
Okay. So speaking of the capillary and the dilation that happens, how reversible is that dilation?
Yeah, so it's actually really interesting because, especially for type 2 diabetes, a lot of people in the field will say that it's reversible, and that to a point, I would say hypertension is, I won't say completely reversible, but you can definitely alleviate your symptoms. And a lot of doctors will tell you exercise, eat a better diet, that kind of thing, which is obviously much easier said than done.
Sure.
So it is, I would say it's definitely something you can do by virtue of just decreasing body mass index or body fat percentage. But again, easier said than done.
Sure. That makes a lot of sense. So are there, so in the experiments that you showed with the rats, you had like normal fat and high fat diets. Are there any other, thinking about sort of applying it to people, sort of, I guess, quote unquote, bad diets would be like high fat, high salt, high sugars.
Yeah.
Do those, are they confounding factors, or is it more like, thinking of like I guess the keto diet, I think, if I remember correctly, is the one where it's like, try and cut out all sugars and then high protein, high fat. Right. And so, like, would you expect to see the same issues show up with that kind of diet, where, like, let's pretend that they could cut out all the salt and all the sugar, and they only had the high fat diet? Would you expect to see the same thing?
So are you asking if I would expect to see the same thing in a high salt or high sugar diet fed mouse as well?
Yeah, I think so. I'm trying to ask it the right way, but yeah, I think that's what I'm asking.
Yeah, so I would think that we would. The only issue would be that I don't know of any studies where people have given mice a high salt, or I don't know if they make a high sugar diet for mice. That would be interesting. But giving mice a high salt diet for, 20, 28 weeks, whatever, I don't know if that's sufficient to make them obese. I can imagine that it would definitely give them hypertension, which again would also lend to, I mean, that's really the interesting and also frustrating thing about this research, is everything is kind of intermixed. So if a mouse becomes hypertensive, it's likely that, of course, they'll have, kidney disease, but they may also become obese as well. And so when we're looking at this particular question that I have, which is really the effect of feeding associated mechanisms on blood pressure control, but I'm also looking at mitochondrial gene expression. We used the high fat diet because we kind of wanted to use this example of intermittent fasting. And so we know that a lot of people will kind of tell someone, hey, you should do the 16-4 split on intermittent fasting. And so we wanted to kind of mirror that in an obese mouse. So to answer the question, I think that we would definitely see some effect in terms of metabolic distress in a high salt or a high sugar fed mouse. But I don't know if it would have the same effect in terms of the time restricted feeding pattern, or if it would just be something that, maybe would take us longer than 2 weeks to see an effect.
Okay, I got you. That's one that always, it always interests me, just because I know there's such a compounding of factors that can happen there. That I was curious about that. Okay. So did you buy for the PCR experiments that you were doing, where you had the multi-well plate? Did you, one of our chatters wanted to know, did you buy a customized plate from Bio-Rad, or did you design your own array?
No, we used, these are from QIAGEN. It's the RT2 profiler array, and we just used the mouse mitochondria plate that they had already pre-made. As I said, they're pretty pricey, so they run about $200 a plate. And again, each plate is one sample, so you have to run the same cDNA in every single well. So I definitely, in a dream world, would love to create a custom, mitochondria or, kidney mitochondria gene associated array. But I don't think my boss would let me swipe the card for that.
Fair. Yeah, that adds up real fast.
Yeah.
When you were showing the regulation of the different genes, you had some that were denoted as up and down regulated. What's the criteria for somebody that's not in the field? How do you determine what's an up and down regulated?
Sure. So we used the normal fat diet fed mice as our control. And so the example would be, the normal fat diet mouse would be the control. So if we're looking at and I'll go back to that figure just to explain it a little bit easier. Okay. So for this example, we're looking at the high fat time restricted feeding mouse at ZT5. So this would be our control in this example. Yeah. And so now we're looking at the time restricted feeding at ZT17 relative to the ZT5. And so what we're looking at here is anything that's unchanged means that there was no significant fold change between ZT17 and ZT5. And so for determining whether a gene is up or down regulated, we looked at whether it had a plus or minus 2-fold change. So was the fluorescence that we get from the PCR, was that 2 times or more above or 2 times or more below the reading that we got for just the ZT5 genes? I don't know if that makes sense. No, but in order to determine if something is upregulated, it's a plus 2-fold or above fold change. Downregulated is a minus 2-fold or below fold change.
Okay, that makes sense. Let's see. So for the kidney, okay, so one of our chatters was wondering, is there a typical cap of brown fat surrounding the kidney that's different in the high fat diet?
That's a really good question. I actually got this question before. There is brown fat that kind of pads the kidney, and that was in the high fat mice. Obviously, they have a lot more fat overall, so there was a good bit more brown fat surrounding their kidneys. But that's to be expected with the high fat diet, especially having been on it for 20 weeks at that point.
Okay. So this one has an acronym that I don't know, but I hope you will. What was your FDR?
FDR. I don't know that one.
All right, let me see. Rama, what's FDR?
I probably should know that one, but I don't.
Oh, false discovery rate. Okay.
False discovery. I don't know.
Let's see. Oh, threshold. The threshold that you use as a cutoff for determining the change in expression.
Oh yeah. So that it was plus or minus 2-fold was our cutoff for upregulated or downregulated.
All right. Yeah, that makes sense. I had a question and it just left me. Oh man. Oh, I know what it was. Again, I'm only passingly familiar with how these experiments are done. And I know certainly the time and financial costs that are involved here may affect this, but how do you or I guess what is your sample size for these, and how do you sort of check for like reproducibility and things like that?
Yeah, so that's one of the pitfalls of this series of experiments. One is it's pretty expensive. So the each of these is run in just one technical replicate. So each well on that microarray plate is, you only have one shot. And so definitely that's something else that's in the future. Hopefully we get a couple of grants approved and we can run these all again.
Sure.
So really it's an N of 1 for all of them. And as far as seeing how we're able to replicate this, in a 96 well plate, it's kind of going to be the same issue as pipetting efficiency and pipetting accuracy. So probably just having the same person do them again and not having someone else do them. So I'll have plenty more cDNA and plates to run in the future. But yeah, this is all an N of 1.
Okay. All right. Did you look at the changes in the ion channel expressions, especially the calcium one?
I didn't, but like I said, that microarray has a number of like SLC25A family genes. So I think that calcium would be an interesting one to look at, and we definitely could do that. It's just a matter of pulling those numbers out of our Excel file and then running the qPCR. But we did not look at any of the other ion channels.
Okay. Rama, this is Dr. Kothapalli. She's a professor from Oklahoma. She wanted to recommend that you think about applying for the Ruth Kirchstein, I hope I pronounced that correctly, award.
Yeah. That's actually part of my qualifying exam that I'm getting ready for, is essentially writing the application for that. Perfect. So I'm currently doing that.
Excellent. Good.
I appreciate the suggestion.
Absolutely. So I'd like to also talk about the policy side of things that you discussed, just in a more general sense. So I'm in South Carolina, you're in Alabama. I'm sure we could share some thoughts about the political winds that exist in our states, especially as it comes to science and whether scientists are trustworthy or not. Could you talk a little bit about, in the face of that kind of, I don't want to say opposition, but that sort of barrier that has to be overcome, in terms of pushing for science policies, that kind of thing. What do you think? What in your opinion is sort of the most effective way to go about doing that?
The most effective thing, and it's also probably the most difficult thing, especially for those of us who are, actively in a lab most of the day. But the most effective thing is always just actually working with the community, actually getting out there and, telling people, hey, especially in the context of the pandemic, was getting out there and telling people, hey, I'm a scientist. I have read these papers, I've looked at the materials and methods and all that. And, based on that, I totally stand behind the suggestion, or in some contexts, I don't stand behind the suggestion. And that was obviously very difficult to do during the pandemic, when people either A, didn't want to come out of their house at all, or B, didn't want to pretend, or didn't want to, face the fact that anything was happening in the world.
Sure.
But I know that for me as a grad student, it's been really helpful just being a part of a lot of different student organizations that have these kind of ends with higher administration, and getting to go to a lot of their like committee meetings and task force meetings, especially related to the pandemic, and just saying like, yeah, this is all a great idea in terms of, the student population, but is there any way that we can also expand this into the community, whether that's working with an organization outside of the university, or even just gearing some of our resources toward the surrounding community. So I know for University of South Carolina, as well as for UAB, we're kind of the downtown area. It's really, really kind of interweave between each other. And so I know that when the pandemic started was that a lot of our houseless community, they didn't have masks, they didn't have hand sanitizer. A lot of them had no idea what was going on. They just woke up one day and nobody's on the street. And so one thing that I was able to do that I was really excited about was just going out with a couple other students and just giving out masks, giving out hand sanitizer, kind of explaining to them, here's what's going on. This is the best way for, you to prevent yourself from getting sick. If you have any of these symptoms, this is what you need to do. But yeah, I think that that's probably the best way of interacting with the public, is to just physically go out there or work with an existing organization that already has a really good rapport with the public, and just kind of make yourself known and make yourself a resource for them.
Really good advice. That's a great way to do it. In your conversations with when you went up to Capitol Hill and actually got to meet with some of the legislators, did you get? Certainly they may have done whatever they were going to do, but did you get a sense that they were taking what you were speaking with them about seriously? Like whether or not they actually followed through, do you feel like that helped as well?
Yeah, absolutely. It really was both exciting. It was really exciting in general, obviously being on Capitol Hill and, seeing the buildings and all that stuff, but
Sure.
also in basically all of the actual politicians as well as their staffers that we spoke to, they all seemed really, gung ho for increasing funding,
Okay.
because, and one of the things that really kind of put it into perspective for me is that a lot of the students that I went with, we were all doing some form of biomedical research. So generally, we're looking at diabetes, Alzheimer's, Parkinson's, cancer, all of these things. And one of the kind of, people that work for ASB&B that was up there with us was like, a lot of them are going to be perfectly fine and very excited that you're in their office, because a lot of them are either suffering from these things, family members suffering from this, they're not getting any younger. These are all diseases that happen as you get older. So it was really a practice in, I'm researching this particular thing. How can I, bridge this gap between what I'm researching and something that's going to mean something to this staffer or this politician? And so everyone seemed really receptive. Everyone seemed really excited. I actually ended up talking to Rick Allen, who's a representative from Georgia, about both NIH funding as well as reforming the K through 12 STEM education in the state of Georgia. And that actually went really well. It was a really kind of like a more personable moment with him, because it turns out he actually went to high school right down the street from where I went to high school from. And his family's his family's construction company built the house that I grew up in. And so I was able to tell him, like, hey, we have all of these connections. I am a scientist who wants to, someday possibly run for office. You have this seat. What are we going to do for, the kids that are coming up behind us that are going through the same schools and, kind of running the same streets that we ran? And he was really receptive to that and actually kind of had his education staffer follow up with me. So I think that it's probably a lot more meaningful to them when you actually show up at their office, because DC is not cheap. The getting around there, getting there, walking around, it's very tiring. So that definitely, everyone was very receptive and very kind when we were there.
That's awesome. That's really good to hear, honestly, because a lot of times it seems like a hopeless thing, but that's good that at least some of them will actually take it seriously. That's really good to hear. Let's see. We've got another question. Going back to the research.
Okay.
You mentioned that your animals were 8 weeks old. Why did you choose that age to put the animals on the high-fat diet?
I chose that age because I knew I was going to put them on the high-fat diet for 16 to 20 weeks, and I didn't want to have to think about some of the other things that can happen with mice as far as gene expression goes when they get, 30, 35, 40 weeks old. So I just chose the youngest age that I could, that the mice were weaned and, big enough for us to be able to collect kidneys from them.
Okay, that makes a lot of sense. So I'll go ahead and get any final questions in. I'll go ahead and start wrapping up a little bit and thank Jazmine again for a fascinating talk, both on the research side and on the policy and advocacy side. Like I said, that's a really cool thing to talk about. There were a lot of good suggestions there. Actually, I appreciate that. That's something I'd like to try and do. Not always very good at communication, but it's heartening to see the work that people are doing to push good science and good policy forward. So I appreciate you doing that and sharing effective ways for other people to do it as well. It's always a great thing to see. It looks like that might be all the questions we've got. So we'll go ahead and finish up. Jazmine, if I could get you to hang around for just 1 minute, we'll talk briefly after this. I'd like to thank everybody for coming out. Like I said, it was a fantastic talk. Appreciate all the great questions. We will be back again next week with, let's see who we've got here. Dr. Manuel Salino. He was actually supposed to present about a month or so ago and had had to go to the emergency room, so we're glad that he is doing better, and he will be back with us to present his work next week. I believe he'll be talking about glaucoma. So that should be another great talk. I hope I'll see everybody out here again next week. In the meantime, enjoy your long weekend. Stay safe. Get vaccinated if you haven't yet. Wear a mask if you haven't yet, and honestly, probably keep wearing masks for a little bit longer until we get a higher percentage vaccinated. I think that's everything I've got. If you have some friends that might want to do that 24-hour stream, links are in the chat. Make sure you let them know about it. We have 15, 30, and 45-minute talk times available, and we'll work with pretty much anything that's science or science related. If there's any questions, feel free to put them in touch with me. I think that's enough rambling. So thank you to everybody for coming out, and we will see you again next week. Have a great weekend.